GeneSketch, Molecular Biology, Simplified. Welcome back to GeneSketch. In our first episode, we established why molecular biology matters, from diagnosing diseases to identifying genetic traits. If it's the ultimate field of applied science, then what is the ultimate molecule at its heart? DNA, the blueprint of life. It's deoxyribonucleic acid, or DNA. DNA is the master plan, the complete set of instructions for building, running, and repairing every living thing. To become confident in the lab, whether you're analyzing a viral genome or a patient's genes, you have to understand the molecule that started it all. What is DNA? Chemically, it's a massive macromolecule made up of carbon, nitrogen, oxygen, phosphorus, and hydrogen atoms. But we don't study the individual atoms, we study the repeating units they assemble into, called nucleotides. Think of nucleotides as the basic, repeating brick in the wall of your genetic code. Every single DNA nucleotide has three components. A phosphate group. A 5-carbon sugar called deoxyribose, the D in DNA. A nitrogen base. The nitrogen bases are the part that holds the information. And there are 4 different types: adenine, A, guanine, G, cytosine, C, and thymine, T. How do these bricks connect to form a strand? The phosphate group of one nucleotide joins with the sugar of the next. This strong chemical bond is called a phosphodiester bond. This phosphate sugar linkage forms the alternating sugar-phosphate backbone of the DNA strand. In the lab, we often talk about the direction of DNA. A nucleic acid chain grows by the attachment of the 5-phosphate group. This gives every strand a natural direction, defined by the numbering of the carbons on the sugar, running from the 5', 5, end to the 3', 3, end. Of course, DNA isn't a single chain, it's 2 strands wound around each other in a beautiful spiral staircase, the double helix. This structure, first described by Watson and Crick, is key to life. 2 rules define this structure. Base pairing, the rungs, the nitrogen bases reach across the gap like rungs on a ladder. They always pair up with their specific partner, who always pairs with T, and G always pairs with C. This is called complementary base pairing. The sequence of one strand dictates the sequence of the other. Antiparallel strands, the handrails. The 2 strands run in opposite directions. If one strand runs 5 to 3 up, the other runs 3 to 5 down. This antiparallel nature is crucial for how enzymes read and copy the DNA. Why does this structure matter? The order of the A, T, C, and G is the code. This sequence information determines everything from the color of your eyes to your susceptibility to disease. So, DNA is the stable, protected blueprint locked in the cell's nucleus. But if it's the blueprint, how does the cell get the instructions out to the protein-making factories? It needs a messenger. Next time on GeneSketch, we move into the dynamic world of the central dogma. How DNA information is copied into RNA, and then translated into proteins. We'll cover mRNA processing, splicing, and the differences between DNA and RNA that make this whole system work. Don't forget to like and subscribe to GeneSketch to build your molecular confidence step by step. We'll see you in the lab.